right, everyone. It's that time when we're supposed to get started. I got some amazingly good news for you. The Indians are going to the World Series. There's that. Yeah, how about the Tribe, huh? But in addition to that, I am not going to talk with you about chemistry anymore. How about that? Now that's got to be good news. We've paid our dues. We've gone through all the trouble of talking about subatomic particles and valences and electronegativity, and then even to those hairy glycolytic and citric acid cycle and Calvin cycle pathways. The point to this part of the class, sort of like an undercurrent that you may not have been picking up on yet, is, and let me just shout it out to you, the point of all of that stuff is that you're going to appreciate in the next couple of weeks that living things are all about the energy, okay? Maximizing the energy that they have, using it in the most efficient way, not squandering it, getting it from other things and other places. It's all about energy. And those pathways that we talked about, photosynthesis and glycolysis and the citric acid cycle, ultimately, those are central to life because energy is such a big deal. That's job number one for most living things, is just marshaling their energy reserves. We got that now. We know how it is that they extract energy. You got the rules of thermodynamics that talk with you about how you have to couple unfavorable reactions to favorable reactions to get them to play out, all that sort of thing, all right? So we got that down. Now I'm going to change gears a little bit. And I'll tell you honestly, there's a rough patch here in my storytelling for you. I feel like I understand the theme, the story. I was building up to glycolysis and photosynthesis in the first part of the class where we've gotten to up till this point. Now the transition shifts not from how you get that energy and how you play around with it, but what you actually do with it, okay? And so where we're headed is I'm going to start talking with you on Tuesday about cell division and reproduction. That's an important thing for a living thing. That's perhaps job number two. Once you've taken care of your energy issues, if you've done a good job, you get rewarded by being able to make more copies of yourself, right? And so that's what's going on Tuesday. But then after that, we get into the real genetics aspect of it, and ultimately I'll be talking with you about how it is that cells turn genes on and turn genes off so as to maximize their ability to play that game with energy. I notice like lights flashing behind me. Is there something going on I don't know about? Good. It's just me. It's the excitement of the Indians going to the World Series, I suppose. So that's where we're at. Is I've sort of closed the book, the chapter, on one part of what I want to talk with you about in this class. And today I'm starting to move in a different direction. And so I said a minute or two ago that I feel sort of like a disconnect. And it's because I'm changing gears, and it's not going to be a particularly smooth transition. Today, though, I'm talking with you about how cells communicate with each other. The reason that they're communicating with each other is ultimately so they can better utilize their energy resources. Okay, so that's how this ties in with what we've been doing up till this point. And it lays the foundation then that if they do a good job with getting that energy in the first place and then communicating with each other about that energy, then they're in a position where they can start to make new copies of themselves. Now you see, I know there's something going on behind me, and I'm not sure why that's happening. But if they do a good job with what we've talked about up till this point, and then what I'm talking with you about today, then they get the reward of being able to make more copies of themselves. So today, again, I'm going to talk with you about cell communication, ultimately with the idea of explaining at some level why it is that we need to coordinate activities. What you see in this picture here, the cheetah chasing the gazelle, that's a wonder to behold, right? If you stop and think for just a minute about what's going on here, it's an amazing set of activities that are playing out in an orchestrated fashion. You can't get a cheetah to run that fast unless hundreds of millions, maybe 1 billion cells are all working in concert with each other. They have to be in sync, right? That motion's not going to happen in a way that's helpful to the cheetah unless you do a heck of a lot of coordinating of activities. And the gazelle, same story. You have to have a lot of coordination of activity to get the gazelle to be able to do what it's trying to accomplish here. At the end of the process, though, all of that coordination is about the gazelle wanting to protect and maintain its own energy supplies, its energy-rich molecules, and the cheetah wanting to get a bunch of energy-rich molecules, those that the gazelle has within it, right? So it's all ultimately about energy, but to be able to preserve that energy or to be able to get that energy, there has to be a lot of coordination. A lot of communication needs to take place between cells. And when it happens right, it's a wonder to behold, right? Just some remarkable stuff is going on. And ultimately, again, this is all about cell communication. That's looking at it in a very high-level way of talking about it. Of course, that's kind of where we're going to end today. I'm teasing you with the end. Let's start at the beginning and talk about how this plays out at a smaller scale, such as what takes place between bacterial cells. Because again, remember, what we do in biology is we'll often figure stuff out with simple prokaryotic organisms and use those insights to give us ideas about how it is that more complicated organisms are doing their thing. So, would you believe it? Bacteria, simple as they are, bacteria communicate with each other. It's not hard to impress people that the cells in a multicellular organism, and that's code for eukaryote, by the way, right? Eukaryotes are the multicellular organisms. We're also the things that have the internal membrane-bound compartments. But it's not hard to convince people that a eukaryote, a multicellular organism, needs to have its cells communicating with each other. You need to have the muscle cells working in a coordinated fashion with the nerve cells and all the rest. That kind of communication shouldn't be too much of a surprise. But for prokaryotes and archaebacteria, but let's focus on prokaryotes. Prokaryotes are invariably single-celled organisms, okay? They don't form bodies. They don't have hands and teeth and eyes. They're, for goodness sakes, a single cell, complete unto itself, doesn't need anything from any other organism, right, or any other cell. It's a complete package all by itself. But information is a powerful thing. It's something that prokaryotes recognize, apparently, can help them play this game of getting more energy and using energy more efficiently than others. And so they're looking for every possible advantage that they can have in their marshaling of their energy resources. And one of the best things that they can do to make the right decisions in terms of what genes get turned on and what metabolic pathways to have running and which ones to mothball for a little while, one of the best things that they can do to make the right decisions there is tap into information from their surroundings. And what better source of information than cells like yourself that are playing the same game? And so it shouldn't in the end be too surprising that bacteria, particularly bacteria that are part of the same team, right? Like bacteria from the same species. E. coli is a great example, right? Wouldn't it make sense for one E. coli to share information with another E. coli so that both of those E. coli cells could make better decisions about how to use their energy reserves? What they should be doing to most maximize their ability to use and extract energy from their environment. It's especially true because, again, they're on the same team. Prokaryotes can divide in a way, and we'll talk about this a little bit on Tuesday, but prokaryotes can clone themselves. They just make perfect copies of themselves. And so when one E. coli, like a soapbox derby race going on in the halls outside here today. It's a wonder to see that, too. That's great coordinated action. But one E. coli is essentially a perfect copy of its neighboring E. coli. If you help a perfect copy of yourself, that's actually not all that different from just helping yourself. So from a genetics, from an evolutionary perspective, there's another reason for there to be cooperativity, particularly between bacteria that are of the same species, because they're very often perfect copies of themselves. So it makes sense. It's a good idea for bacteria to chat, to swap information. How's it looking over in that part of the test tube? How's it looking in this part of the test tube? It's good for them to be able to do that. How do they pull that off? Well, this slide that's been hanging behind me for a couple minutes now gives you one way that bacteria communicate with each other. One thing that's important for bacteria to know is to just have a sense for how many other bacteria are around. You can imagine that if you are alone in a room, you might make a different set of choices than if the room is crowded with a bunch of other people, right? You pick the scenario. Do you eat the pizza now or do you wait till later, right? That might be one example where if you're alone in the room, you might have a different course of action than if the room was full of people, right? If the room's full of people, I might be going over to get the pizza right now because I'm afraid it's not going to be there later. If I'm the only person in the room and I'm not that hungry right now, I'll wait, right? You know, maybe a little later I'll go back and I'll pick up the pizza at that point. So knowing how many other bacteria are around is something that bacteria find very useful information. Very useful information in terms of how it is that they want to use their energy. Well, the way that one bacteria knows that there are other bacteria like itself around is by producing signal proteins. So the picture there, it's a pretty good cartoon rendition of an E. coli bacteria, that tube-like thing. To the left, you see a bacteria over here. A bacteria will do this thing called constitutive expression of a signal molecule, typically a small protein. I used a fancy word there. I said constitutive, or we could talk about constitutively. Constitutive, and we'll get into this more when I'm talking with you about gene expression, but constitutive is just fancy genetics talk, molecular biology talk for a gene being turned on all the time, regardless of the circumstances, right? This is just something that's a protein that's getting made at a low level all the time. Background noise kind of stuff, if you like. It's just always there. And what these bacteria will do is they will constitutively make these signals. They'll constitutively make these signal molecules, and they'll have on their surface a receptor that can bind to those signal molecules. And what will happen is the more bacteria that are present, the greater the concentration of bacteria, the greater the concentration of the signal molecules, and at some point, a threshold gets crossed. The bacteria starts to realize, hey, there's a lot of these signal molecules here. That must mean that there are a lot of these bacteria here of my own kind, right, because they're communicating to themselves. And what will happen is once that threshold gets crossed, once there's a sufficient number of receptors on the surface of the bacteria that are binding to those signals, a message gets communicated to the bacteria's genes that says, okay, change of course, a little bit different way to grow right now. There's a crowd. There's a bunch of people in the room. We need to behave a little bit differently than we did before that threshold got crossed, right? So it's a fairly simple system. It took me a lot of words to say this, but it's really a pretty simple system. You got bacteria that are sending out a little signal saying, I'm here, right? When a lot of those bacteria are saying, I'm here, they hear there's a lot of bacteria here. When they get to a certain point, they say, okay, there's enough bacteria here that I need to change my strategy, the way that I'm growing, the way that things are working inside of me. The picture here, again, I said it looked sort of like E. coli. here's where the rubber hits the road. I'll give you a specific example for E. coli. E. coli grows in one of two ways. It either grows, its metabolism is set in either one of two positions. It either grows in log phase or lag phase. Log phase is what's happening when the E. coli has got the pedal to the metal. Crank it out, let's do everything we can, grow full tilt, metabolism cranked to the max to make as much volume, more copies of the stuff inside of our cells so that we can divide and make 2 copies of the E. coli where once there was 1. Full tilt. Metabolism going as fast as it can with the ambition of increasing the mass of the cell by making copies of all of its proteins and its DNA and its lipids, so that that big cell can divide into 2 smaller cells. Logarithmic growth. Every 30 minutes, you double the number of E. coli that are present. That's log growth. The other choice that E. coli has is lag growth, or lag phase. And what happens there is the E. coli just shuts everything down, right? The metabolism, it's night and day, right? Take it easy. There's a lot of other bacteria hanging out here. So that's the play-by-play guy for the soapbox derby. They come a little bit after the lead cars go by. What happens is they just shut things down, and they hunker down, and they say, okay, there's a crowd. Times are a little bit tougher, perhaps, than they were before. There's so many that it's probably not a good idea for us to divide anymore. Our baby cells aren't going to have a very good chance to survive. Hunker down. Tough it out, hope that times get better and that the crowd disperses at some point. Maybe the other bacteria here are going to die. Maybe they're going to get diffused out into other parts of the environment. But when things get better, then we can switch over. But for now, just hunker down, keep the engine running on idle, more or less, but no more of this full tilt, making more proteins, more lipids, more DNA. Just keep the fire burning, nothing fancy. One of 2 modes of growth for E. coli, log growth or lag phase, right? When there's a crowd, what happens is the cells turn green, like they do in this picture here, or at least as they do in this picture here. They behave differently. It's a night and day kind of difference. They perceive a certain set of circumstances in their environment, their surroundings, and they respond accordingly. And when the circumstances change, they can switch back to the log growth phase, and they can go full tilt and double their number every 30 minutes. It turns out that that's a really important strategy for E. coli to be able to do. And they do it, and we'll talk about exactly how they make that switch in about a week and a half, maybe 2 weeks' time. Okay? But so there's one kind of signaling. It happens even with things as simple as bacteria. And I'll tell you, in your lecture notes, I also slip in a story that just fascinates me, but another example of bacteria communicating. What we have here is bacteria communicating with other bacteria of the same species. I give you an example in the lecture notes about what's going on inside of your mouths right now. I said inside your mouths right now. Right? If you haven't seen this part of the lecture notes yet, it's kind of gross. But there are thousands, like 25,000 different kinds of bacteria living inside of all of our mouths right now. I don't care how recently you've been to the dentist. I don't care how thoroughly you brush your teeth and what kind of mouthwash you use, right? We all lug around about 25,000 different species of bacteria in our mouths at any given point in time. And about 1,500 of those bacteria are what are involved in making what dentists call dental plaque, the thin film, sometimes for some people thick films, of bacteria that live on in between our teeth. All right? Lots of different species of bacteria. How about this? Isn't it? I marvel at the fact that these different bacteria, a large fraction of them, work in a symbiotic way. They interact with each other. They help each other out. They get some benefit from the interaction. The thing that's interacting with them gets some benefit. It's a mutual symbiotic relationship that starts to rival the kind of complexity that you can get from a multicellular organism. Even though they're different species, these different cells can cooperate with each other in a way that starts to rival what you see with a multicellular organism. I probably haven't said this plainly enough to this point. It's going to become clearer as we move along through the class. But here's the big advantage you get from having internal membrane-bound compartments. Here's the big advantage you get from being a multicellular organism. That complexity lets you do stuff that you couldn't have done if you weren't so complicated. It's pretty much as simple as that. And prokaryotes are hobbled in that they don't have the internal membrane-bound compartments, and they don't have different kinds of E. coli. It's not like there's a hand E. coli and an eyeball E. coli. There's just E. coli. But what happens in our dental plaque starts to get to the point where we're talking about what you might have been able to do with a multicellular organism, even though we're only talking about single-celled organisms. So these bacteria living in our teeth, on our teeth, you know what they're doing? They set up, they organize in layers and in particular arrangements that are very reproducible, right? Not just within my mouth, but within your mouths as well. They actually hook up together in particular ways. One kind of cell will interact with and attach to and anchor down to another type of cell. Hundreds, thousands of them doing this in a fairly organized way, very predictable, fairly typical way from one individual to another. And what you end up seeing are structures that look sort of like multicellular organisms. They'll have channels within them that are lined by a particular type of cell, that have cilia on their surface, that can beat in a coordinated fashion to get waste products from deep inside of this layer to outside. And they have other types of bacteria that are oriented and positioned and set up in such a way to help bring food particles into this film, to help those bacteria grow. A lot of the bacteria that live in our mouth are anaerobic bacteria. This is a great time to talk about being anaerobic, right? Right on the heels of talking about oxidative phosphorylation and such. Anaerobic means they grow, but only in the absence of oxygen. They can't handle oxygen. A lot of the bacteria in our mouths are anaerobic. How can that be? There's oxygen coming in and out of our mouths all the time. What's the deal there? You know what the deal is those guys take up positions in the middle of the plaque layer, and they let the aerobic bacteria be on the outside to suck up the oxygen, to use the oxygen to drive certain chemical reactions. And then the anaerobic guys, deeper on in, are set up to be able to metabolize things in a way that works best in the absence of oxygen and in ways that might not be compatible with the presence of oxygen. And they share products. You know, they may be good at making the amino acid tyrosine, and other bacteria might be good at making the amino acid glycine. They swap back and forth, a very cooperative sort of arrangement that can only take place because of these types of intracellular interactions, that type of communication. So if your dentist hasn't impressed this upon you enough, flossing, it turns out, is a good idea, because what's happening is you physically disrupt those layers of film, and these bacteria have not adapted yet very well to the physical disruption that comes from a piece of dental floss. So that gives us a little leg up on them that they haven't quite figured out how to deal with yet. All right, let's get to something a little bit more interesting, perhaps sex, right? Instead of talking about gross dental plaque, let's talk about mating. Did you guys know that eukaryotes have sex? That's how we reproduce. Yeast is the simplest of all eukaryotes, okay? Yeast, single-celled eukaryotic organism. It's unusual that way, but it's eukaryotic. It has those internal membrane-bound compartments. Like all other eukaryotes, yeast, even though it's single-celled, reproduces through sexual reproduction. Excuse me. There are two different kinds of yeast cells, right? two different kinds of people, male and female, two different kinds of yeast cells. For whatever reason, the folks who characterized this didn't bother calling one male and the other female. The two different kinds of yeast are A-type yeast cells, lowercase letter a, and alpha-type yeast cells, lowercase Greek letter alpha. So two different types of cells, and when a A-type yeast cell gets that glimmer in its eye that it's time to make a baby yeast cell, it goes out into the world looking for an alpha-type yeast cell. And if the two yeast cells, fall in love and get to know each other and go through years and maybe decades of dating, in the case of my daughters, what I'm sure is going to happen, if the time is right, the two of them will come together. And they'll make something called, and this is the technical term for it, I'm not making this up, they'll make something called a shmoo, S-C-H-M-O-O, that's a little extension of the cytoplasm from both of the cells. They shmoo toward each other, the shmoos fuse, and you end up having both of the yeast cells become one, and holy matrimony, I suppose, and then they swap some of their genetic information, and they break up the shmoo, and they go off on their separate ways with different sets of genetic instructions than they had before. So what we've got here, ultimately, is another kind of cellular communication. Here at a eukaryotic level, what's really happening at a molecular level is the A-type cells are making proteins that get secreted out into their environment that get received by receptors on the surface of the alpha-type cells. And at the same time, the alpha-type cells are secreting proteins that are bound specifically by receptors on the surface of the A-type cells. That's what causes the shmoos to form and to move in the right direction. They're going down that concentration gradient. And that ultimately results in successful mating of those two different kinds of yeast cells. Now, just as an aside, not necessarily directly involved with communication, but notice there's communication that had to take place here. And I think maybe what I need to do right now is just point out to you, did you see how that communication took place? It's a two-part process, really. One cell's releasing a signal, and the other cell's got a receptor that can stick to that signal specifically, right? No point in releasing a message if there's no recipient for that message, right? And you want to have some specificity with the things that are receiving the message. You don't want to just hear that there's something going on. You want to know what's going on. Those receptors are specific. So that's the big picture from this slide. But as an aside, let me tell you this. Yeast can switch its mating type, right? Sort of like a gender change kind of thing for yeast. There's a cassette of DNA that's on a yeast chromosome that if it's in one orientation, it causes the cell to make the alpha protein and the A receptor. And if it's in the other orientation, this little cassette of DNA, if it switches its direction, it ends up making the A receptor and the alpha protein, or the opposite of whatever I said the first time around. Okay? So it's just a matter of a cassette being switched, and that's what it's called. It's cassette switching for mating type switching. And that, too, is something that involves communication, because if a yeast cell finds itself, say it's an A-type yeast cell, if it finds itself hanging out in a place where all it ever sees are the A signal and never any of the alpha signal, it gets the bright idea saying, hey, look, I'm at a party with a bunch of guys. Maybe if I switched over to being female, I'll get some good attention here from this party, and it makes the switch. Right? So there's a sensing that's going on by the A cells in the alpha cells that are monitoring the relative concentration of the different kinds of A and alpha protein that are present. And if it finds that it's in the class that's making the protein that all the other yeast cells are making, it realizes this is a good chance for me to change sides and go to the other team and become, if I'm an A, now it's time for me to become an alpha. So they can actually monitor the concentrations of their own protein to see if it might not be to their advantage to switch. And they switch by flipping that cassette. All right, so communication, microorganisms. That's pretty much what we've been talking about so far. Moving along into eukaryotes like yeast, still single cellular organism. Let's get to something a little bit more sophisticated, like what's taking place inside of multicellular organisms, things like you and I. So, at a very simple level, you can have communication between one cell and another cell in a eukaryotic organism simply by having molecules physically pass through holes in the membrane into another cell, right? Wouldn't be a complete story if I didn't point out, yeah, that can happen. Yeah, that happens a lot. Between cells in our bodies that are physically right next to each other, there will be pores, holes in their cell membrane, and there's the opportunity for molecules to pass wholesale from one cell to the other. Pretty effective means of getting information about the circumstances of the one cell to its neighbor cells. So you can do that. Happens in plants, happens in animals, and in animals, we call those holes, that sort of fusion between the two cells, a gap junction. In plant cells, it's a plasmodesmata. Again, we're just talking here about holes that allow molecules to pass between one eukaryotic cell and another. All right. Then, there's the kind of signaling that takes place without physical connections between the cells, but just because the cells are in close proximity to each other, things that we could call local signaling, like you might have taking place between two adjacent nerve cells. So here we have a picture of an axon that's releasing molecules that are going to trip some trigger on the surface of this cell and communicate some information that way. But just as that can work over short distances, you can have the same sort of thing taking place over long distances. So you could have local signaling by having one cell release proteins or signals that get picked up by an adjoining cell, or you can take this on a larger scale and you can have a cell release some molecules that get distributed throughout the organism and communicate to cells across a very great distance. The example on the right is something along the lines of what might happen with insulin being released from your pancreas and tickling cells throughout your body, telling them about ways that they might want to be metabolizing glucose at that particular point in time. This is more of the sort of thing that takes place on the left side of the cell between one nerve cell and another nerve cell. So we can have that kind of communication between cells. Let's take a little bit of time to talk about what's actually playing out at the molecular level. Just as within prokaryotes, what's got to happen is you need to have some sort of signaling molecule. There's going to be something in the cell's membrane that receives that signal. Okay? Important part of the process is reception. Notice in this picture here, the signaling molecule has a particular shape, and that the receptor molecule has something on its surface that provides a nice snug fit for something with that shape. And I hope that you guys at this point appreciate it's not just the shape that's important, but there should be complementary charges, right? So if there's something negative on the red thing here, there should be something positive on the receptor here, right? A hand-in-glove kind of fit. It's not just the shape, but it should also be something that makes the receptor and the signaling molecule comfortable in terms of hydrophobicity and charge. But so a receptor comes and it finds a signal molecule comes and it finds a receptor on a cell's membrane. Then the presence of that signal molecule needs to be transmitted to inside the cell. and typically needs to be transmitted not just to inside the cell, but also to the cell's DNA, because the DNA is the brains of the outfit, it's the blueprints. A lot of times when you change, when the cell perceives a change in its environment, it needs to change the genes that are being expressed, which genes are turned on and which genes are turned off. And so the signal needs to be communicated from the outside of the cell to the inside of the cell. And very often that will happen by having the receptor molecule interact with some interior molecules, some proteins, for instance, that are water-soluble in the cytoplasm of the cell that can't interact with the signal molecule directly because, again, these are water-soluble. This is a lipid bilayer. This protein here may have amino acids on its surface that allows it to be an integral membrane protein. It can hang out here, but not this guy. And so the signal gets relayed. When the signal binds to the receptor changes its shape in some important way. It's an allosteric effect. And that change in shape causes this receptor protein to interact with a protein on the interior of the cell. That protein might get activated, and in turn activate another protein, and that in turn may activate another protein, and we can have a ripple effect going through the cell, communicating that this particular signal has arrived. So you get reception, and there's a series of steps that we could call transduction, and then ultimately that should result in a response. So for communication between eukaryotic cells to work, it's a three-step process. You have to have the arrival of the signal and the recognition of that signal. You need to transmit that information to the cell, and then you ultimately need to have the cell respond to that information, have some change that occurs because that information has been received. So in this last slide, it all begins with this receptor, right? Sort of good news for you. Cell biologists have been beating up eukaryotic cells for decades now, trying to characterize the kinds of proteins that act as receptors. And the good news is that almost all receptors fall into one of just three categories. There are the G-coupled proteins, there are the tyrosine kinase receptors, and the ion channels. And everything else seems to be a footnote. So within eukaryotic organisms, multicellular eukaryotic organisms, that first step in the process, the signal recognition, and then the passing of that information to the inside of the cell, is accomplished by one of just three different kinds of proteins. And this is actually the order, it seems, in which they're abundant, right? These are the most abundant, the G protein coupled receptors. Next most common would be the tyrosine kinase, and then last would be the ion channels. So let's take them one by one, and I'll tell you what it is that each of them do and give you a sense of how it is that they work. Here is a cartoon rendition of a G-coupled protein receptor. There are a couple things that I think you can pull out of looking at this picture that you don't need me to tell you about, but I'll put some words to it anyway. One of the things that I think it's worth noting is, check it out, this is an integral membrane protein. It's actually embedded in the phospholipid bilayer. The receptor, no big surprise, it's what we saw in the previous slide as well, 2 slides ago. The receptor is membrane bound. How did it get to be membrane bound? Well, it's got these alpha helices that are part of its structure that display on their surface amino acids that are hydrophobic, that like to hang out in the lipidy part of the phospholipid bilayer. So along this edge, there should be hydrophobic amino acids. Right up here at this part of the alpha helix, there might be some amino acids that are positively charged to anchor it next to these negatively charged phosphate groups. Ditto for this end. The alpha helix here may have a couple of positively charged amino acids to make it comfortable hanging out there. Inside of the alpha helices, so from one alpha helix to the next, They could be hydrophobic interactions that cause this alpha helix to stick here and this one to hang out there, or it could be that there's some complementary charges that are causing this alpha helix to like to match up to this alpha helix. All right, so there's that level. These are integral membrane proteins. The amino acids that you find on the surface, and these tend to typically be alpha helices, they tend to be things that would cause it to be anchored in the membrane. There's an important orientation to these signaling molecules. There's no point, it's a waste of making the protein if you're a signal receptor protein and the receptor part of you is pointing to the inside of the cell, right? The orientation is important. So cells need to pay attention or take care to make sure that when they're synthesizing these membrane-bound proteins, just as a reminder, that would be in the rough endoplasmic reticulum. When they're being synthesized, when they're being translated in the rough endoplasmic reticulum, the orientation is important. Not only does it have to be membrane-bound, but the receptor binding site for the signal molecule needs to be on the outside of the membrane as opposed to the inside. And so, two things. Membrane-bound, the signal part, the binding site needs to be pointing to the outside of the cell. And on the inside of the cell, there needs to be some part of the protein that is set up so that it can interact with G protein. Right? That's why this is called a G protein coupled receptor. We're talking here, what we're looking at on this screen right now is the receptor. It's going to get coupled to another protein called G protein. G protein needs to be able to find some features on the surface of this protein here, this inner surface of the membrane, that say, this is one of those proteins that I interact with. What happens when a signal comes along? Well, you get that hand-in-glove kind of thing, the interaction that takes place here. The binding of the signal causes some change in the shape, allosterism. Change in the shape changes the function. That change in shape here needs to be communicated somehow throughout the protein, at least to this part, so that the shape of the part that's interacting with G protein changes shape as well. Okay? And funny thing about G-coupled protein receptors, I hesitate to mention this to you. So I'm afraid you might get the wrong idea. But experiments have been done that show the parts are interchangeable. And what I mean by that is that you've got a specific signal molecule's receptor here, right? So say it's to molecule A, that's what binds here. If you swap out this part of the protein with the equivalent part from another G-coupled protein receptor, one that binds specifically to B protein, what you do is you change the function of this receptor so that it is no longer specific for A, it's specific for B. How about that? So the basic design here is pretty much the same for all G-coupled protein receptors. The thing that's an important difference is typically what's going on with the signal binding site. So again, something of an aside, but for all intents and purposes, you just need to be membrane bound. Anything that gets you to be membrane bound will get the job done here. The business part of the molecule is the differences that you find in that signaling molecule binding site. So that's what a G-coupled protein receptor looks like. Let's talk a little bit about what it is that happens when one of those receptors binds its specific signal. Different kind of cartoon here. The protein we had been looking at on the previous slide now is shown as something like this. You can see it's embedded in the membrane, right? On one side is the signal receptor spot, the active site, if you like, of this particular protein. And on the other side is what specifically will interact with G protein, but only interact with G protein if the signal is present. Okay, so that's the player here. Here is a picture of G protein. You can see G protein is shown now in an inactive form. It's got GDP associated with it, guanine diphosphate, as opposed to GTP or ADP or ATP. It's the same player. But what happens is once the signaling molecule tickles the receptor, there's an allosteric effect that changes this surface of the protein as well, makes it appealing for G protein with its inactive form, the GDP, to come and hang out. That, in turn, activates the G protein. The GDP gets replaced with a GTP, and we now have an active G protein. And what has to happen now is the active G protein needs to transmit that information to other molecules like this enzyme, which here is shown as being inactive, but when G protein hits it in just the right way, it activates the enzyme and it causes you to have a particular response. After the response is complete, the G protein becomes inactive again. You release that inorganic phosphate, and you're ready to start the whole process over again. This picture is really no different than this one. So that's what's going on with G-coupled protein receptors. You know, if I gave you a Cliff Notes sort of summary of what's played out here, G-coupled protein receptors are membrane-bound proteins. On their surface, they've got an active site of sorts that binds to a specific signal molecule. When the signal molecule binds, there's an allosteric effect, a change in the shape and then the function of the receptor protein. That change, in turn, allows G protein on the inside of the cell's membrane to be activated. The activated G protein then goes and kicks something else and elicits a response to the presence of that signal. That's it in a nutshell. So that's how things work with G-coupled protein receptors. One down, two to go. All right? Next on our list of these receptor proteins are things called tyrosine kinases. I don't think there should be too much mystery to what these things are based on their name, right? You guys know full well right now that if kinase is part of the name of a protein or an enzyme, that part of what this thing does is it moves phosphate groups around, all right? That's what we meant in glycolysis and the Calvin cycle. That's what we mean for tyrosine kinases. And further, these are tyrosine kinases. You know what's getting the phosphate group? Ta-da! It's the tyrosine. Tyrosine is one of the 20 different amino acids. It can be phosphorylated. So you can attach a phosphate group directly to that R group of the amino acid tyrosine. All right? Hence, tyrosine kinase. Here's the way tyrosine kinases work. They're membrane bound. See, got a membrane here. See, there's a part of the protein, the tyrosine kinase, that is embedded in the membrane. It's an integral membrane-bound protein. All right? Part of the protein extends outside of the membrane. Typically, a big part of the protein dangles on the inside of the membrane. But it's a membrane-bound protein because of what's going on over here. The part that pokes out has sort of an active site. You get that hand-in-glove kind of fit between a signaling molecule. If you want to be fancy, you could call it a ligand. It's a signaling molecule. You get this hand-in-glove kind of fit between the signaling molecule and something on the outer surface of the tyrosine kinase. When that happens, you have an allosteric effect. You change the shape of this protein in a particular way that makes it actually more amenable to interacting with other tyrosine kinases. And when you get two tyrosine kinases that both get tickled by the signal molecule and they come together, the tyrosine kinase doesn't act as a solo player anymore. It forms a thing called a dimer. And that just means that there's two of them there. Two copies of the same protein. Here, both of those copies have been tickled by the signal molecule to change their shape in a way that causes them to want to come together. It's as simple as that. When the tyrosine kinase is in this dimeric form, when two of them are together like that, they can kinase each other. They can phosphorylate each other. So one puts phosphate groups on the other, and I'll scratch your back, I'll phosphorylate you, you phosphorylate me. Everybody gets happy here, because what ends up happening is there will be, and this is the way it is for all tyrosine kinase receptors, the only thing that differs is what the receptor is up here and what happens at this other end down here in terms of what other molecules that these tyrosine kinase receptors interact with. But what happens is that 3 tyrosines on one of the monomers will be phosphorylated by the other monomer and vice versa. You'll get 6 phosphorylations when the receptor molecule is bound to its signaling molecule. I hope it's not a surprise at all to hear that if you put 6 phosphate groups on this dimer, that extra negative charge is going to have some important changes, going to cause some important changes to the shape of the molecule. Those changes in the shape will change function. And what can happen then is that these other proteins now, because of the change in the shape and the change in the charge of the activated tyrosine kinase, can in turn be activated and elicit some responses inside of the cell. All right? It's that same 3-step process that we saw several slides ago. And with the G-coupled protein receptors, the signal molecule binds. There's a change in the shape of the receptor protein, and that change in turn causes a signal to be transmitted to some other proteins here, proteins that are inside the cell. Let me pause and just point out, this signal molecule never made it inside the cell, right? That's the way these things usually work out. The signal molecules are usually water soluble, and it's hard for them to punch their way through that cell membrane because of the hydrophobic part of the phospholipid bilayer. Instead, the way the information comes, it's not because the signal molecule moves through, it's because you change something in an integral membrane-bound protein. All right, last, sort of least, but still very important. There's the third kind, these ion gate channels. So this slide shows you in one fell swoop the whole picture. All right, you get a signaling, let's start with part 1 on the left. You get a signaling molecule that comes and binds to an active site on the surface of this protein. Again, an integral membrane-bound protein. So the signal molecule comes. When the signal molecule comes, hand and glove kind of fit, that kind of specificity. There's a change in the shape of the receptor protein. You can see the way they've drawn it for you here is there was a flap of the protein that had been covering a hole in this protein. But when the signal molecule is there, the flap says, ooh, look at that, that feels good. And the flap comes up and it starts interacting with things on the surface of the signal molecule. When the flap comes up, the gate opens, and you can start to have flow of ions, small molecules, down a concentration gradient. Okay? There were a lot of these green ball ions up here, none in here. There's a concentration gradient, right? You're just going to diffuse down the concentration gradient as soon as that channel is open and available for those ions to pass through. At some later point in time, if the signal molecule falls away, the flap comes back, covers up the channel, and you stop with the flow of the ions. Not particularly challenging stuff, but how about that? We've seen now 3 different kinds of strategies for communicating information from outside the cell to inside the cell. Kind of neat. They all make sense. Nothing too earth-shattering about any of them, but this is actually how that information gets transmitted. All right, so Those are the plasma membrane receptors. Let's talk a little bit about what's going on the inside. You know, the signaling molecules I've talked about so far have been hydrophilic signals, right? I suggested just in the last slide, see the signal doesn't get through the membrane. Not all signaling molecules are hydrophilic. Some of them are hydrophobic. Testosterone, for instance, is something you've probably all heard of. It's a steroid. It's a hormone. Steroids are fatty. They're actually hydrophobic, and they have no trouble moving through a phospholipid bilayer. When testosterone gets released by some of your cells and interacts with other cells, the testosterone can move right past that phospholipid bilayer and tickle a receptor protein that in turn can make its way to the DNA and cause a change in the expression of genes because this protein is influencing the way that genes are being turned on and turned off. So here what's happened is the receptor hooks up with a fat-soluble signal, and that in turn can come and now, in this conformation, interact with DNA, whereas in this conformation, wasn't that interested in binding to DNA. And then ultimately result in a different set of genes being expressed that in turn give rise to a different set of proteins being expressed. Here's something that I think is pretty cool. You might be wondering what's up. There's this idea of a cascade. What's up with these cascades? Why can't the signal molecule tickle a receptor protein and that be the end of it? Why do you need to have the signal molecule tickle the receptor and then it tickle something else and then that thing that's been tickled tickle something else and that thing tickle another thing and that thing tickle another thing? Why do you have to have these kinds of cascades? Why do you have to have these kinds of chains? This is very common for G-coupled protein receptors. This sort of thing happens with all of these transmembrane communications. You've got this thing, a phosphorylation cascade. It's not enough that you activate one kinase, you activate a chain of them. Why couldn't you have just gotten the job done with the one initial interaction between the membrane-bound receptor and an internal kinase? Here is the answer to that question. What these cascades allow, the fact that you don't have just one transmission of the signal, but you transmit the signal to something that transmits it to other things, that also transmits it to other things, those cascades, that path, those number of different proteins that get activated, result plainly in an amplification of the signal. One signal molecule tickling one receptor on the surface of a cell can result in thousands of proteins being modified on the inside of the cell. What's happening here is you can see epinephrine is binding to a G-coupled protein receptor. A specific example, that G-coupled protein, the G protein gets activated, and that one epinephrine molecule may be enough to cause the G protein to activate 100 G proteins. But those 100 G proteins activate 100 adenyl cyclase proteins. And the 100 adenyl cyclase proteins each activate 100 cyclic AMP molecules. And those 10,000 cyclic AMP molecules can activate 10,000 protein kinase A's, and those in turn can activate 100,000 phosphorylase kinases, and you can end up with, what's the math here, 100 million glycogen molecules being metabolized into glucose as a result of that one epinephrine molecule binding. Let me put it to you simply, all right? A lot of times when these signal molecules transmit information to the inside of the cell, they do it in relatively small numbers. There'll be one or two signal proteins, receptor proteins, on the cell's membrane. That, in turn, that signal gets amplified by being run through a cascade, so that a number of different molecules get activated at each step in the cascade, so that you can have one molecule result in this instance, 100 million molecules being affected at the end of this cascade. I go often to biology department talks. Did you guys realize that once a week, typically Mondays at 12:30, the biology department brings somebody in from some other university to talk about their research? I find those talks come into one of three categories. There's the ecology sort of category, where they're talking about how organisms are interacting with their environment, predator, prey, that sort of stuff. Cool. There are the molecular biology and genetics talks, where they're talking about population genetics and how genes get turned on and turned off. And there are the cell biology talks. Cell biologists seem to feel it's important to intimidate the people in the audience with ridiculously complicated cascades like this. And I find myself sort of pitying these folks, because what they have to do is they spend every waking hour thinking about how these very long and very detailed cascades play out. And they have to realize that there can be crosstalk between one cascade and another cascade. It's a tangled mess of spaghetti when you start to look at these maps of how it is that one signal will result in a certain set of actions, and how there can be crosstalk between one receptor's cascade and another receptor's cascade. It's nasty stuff, but nonetheless, people are working really hard and long to figure out how it is all of these cascades work. And there's lots of them. There are hundreds of them, it seems, inside of our cells. They're, again, all sort of woven together into a web. So those are some phosphorylation cascades. You may have noticed that cyclic AMP got made. I just want to give you a flavor for what's going on with cyclic AMP. G proteins often activate adenyl cyclase. Adenyl cyclase makes something called a second messenger. This is the first messenger, the signal molecule. The messenger inside the cell here is cyclic AMP. Cyclic AMP is something that you're going to hear a number of times yet during the course of this class. It comes up a lot in physiology types of classes. Cyclic AMP is a very common, very widely used by cells messenger molecule. During glycolysis, I mentioned to you that it's AMP that comes back in a feedback loop to tell the enzymes at the beginning of glycolysis, crank it up, energy levels are low. That was an instance of signaling taking place, that type of communication from the end of the pathway to the beginning of the pathway. That communication was actually accomplished through cyclic AMP. So here's the deal with cyclic AMP. You start out with ATP, you get yourself some ADP as the energy is getting used. The ADP can be converted to AMP, and the AMP can be converted by adenyl cyclase into cyclic AMP. And cyclic AMP then sends messages to all sorts of proteins inside of the cell. Here they're showing you a protein kinase that it's interacting with. Just to take the mystery away, I don't expect you to memorize this. I just want you to realize, hey, we are talking about actual structures, and the names kind of make sense. This is what ATP looks like. You got your ribose, it's a nucleotide, right? You got your ribose sugar here. Attached to the ribose sugar is a nitrogenous base. This particular nitrogenous base is adenine. And also attached to the ribose sugar are three phosphates. A, adenine, T, P, 3-triphosphate, all right? And what happens with ATP is adenyl cyclase can lop off two of those phosphate groups, and then a very energetically favorable reaction, right? And then forge a covalent bond between the phosphate group and another carbon on the ribose sugar. So that's why this is cyclic AMP. Look at that. The phosphate group is attached to this carbon and then forms a little circle and gets attached to this carbon as well. Just for fun, let's see what carbons are involved with this circle. This is carbon number 1. They get numbered clockwise going around the sugar. That's carbon 1, 2, 3. There's 4, and that's 5. So there's a linkage here between the 3 carbon back onto the 5 carbon of this ribose sugar. Just so you see, oh, it is sort of like a circle. That's why they call it cyclic AMP. Oh, and you can go back to getting rid of the phosphate, that cyclical part. You can break that covalent bond. This one here, you can break it and get back to AMP again pretty easily. All right. I'm going to go through these fairly quickly. Inositol triphosphate, DAG, and calcium are also signaling molecules akin in many ways to cyclic AMP. Cyclic AMP is a molecule that cells use to communicate to a pretty large number of proteins. These 3 guys get used to communicate to a large number of proteins as well. It's kind of like what I was talking about with the lag phase and the log growth phase for E. coli. You're changing the activity of a whole bunch of different proteins when cyclic AMP is involved. You change the activity of a whole bunch of different proteins when any of these 3 kinds of intracellular communicators are involved. Let's talk about what's going on with calcium ions. Cells have unusual distribution of calcium. Right? Outside the cell, you'll typically have higher calcium concentrations than inside the cell, but there are compartments inside the cell, like the endoplasmic reticulum, that are often high in calcium concentration. So there will be proteins that pump calcium in. There will be proteins that pump calcium out. And you can have a G-coupled protein receptor activate G protein that in turn releases inositol triphosphate, IP3, and the IP3 then can, inside the cell, open up a gate that allows calcium ions to move from one location to another. And you can see, as a result of this particular G-coupled protein interacting with this G protein interacting with this particular enzyme, you release this molecule from the surface of the cell, and that molecule interacts with this molecule, and this molecule allows calcium to go from one compartment to another compartment. You're increasing the calcium ion concentration here as a result of what happened way back over here. A change in calcium ion concentration can easily result in the change of the activity of a bunch of enzymes that are present inside of the cell. Hence, various proteins might be activated. And this signal then, in turn, can elicit a particular response. And I tell you what, I think I've said that all I care to say to you guys about cell communication. For sure, at the end of the semester, remember, 10, 12 minutes early on this particular day. This last slide, just if you're curious about some more of the details, it's just making the point that different receptors can cause different responses. Here's the deal. You guys should go and have a good weekend, and I'll see you on Tuesday. And Tuesday, I get to talk with you about cell division. I can't wait. So see you guys then.